Hear me? No? How about no? Yes? No? Yeah? All right. So thanks for coming. This is today is not just a physics colloquium. It's also the joint seminar series with structural and quantitative biology, and we're delighted to have Markus Meister here today from Caltech. He's a professor of biological sciences at Caltech. He studied physics at the Technical University of Munich in Germany, and then did his PhD at Caltech. In physics under supervision of Howard Berg. He then took a postdoc with Dennis Baylor at Stanford University back in '91, and that's when I and many other people in systems neuroscience at the time found out about his work, because he developed a multi-electrode recording array, which allowed him and then later others to be able to record from many cells at once in the retina. And a whole bunch of questions came up at that time that weren't accessible before. Being able to see correlations between neurons, how populations of cells represented a stimulus, and that was a real breakthrough. And in fact, it had generated quite a lot of heated debates even. It was a really exciting time, and lots of people got interested in addressing questions about population coding and asking questions about how interactions between neurons are important for coding. Markus has had, and I should say he started his own lab then at Harvard, continuing work mostly in the retina, but some other things too. And he's had many interesting results. I'll just give you a flavor for one of the ones that made a big impact on the way I think about early processing in the visual system. He found that with Zach Pitkow, actually, I was thinking of this result with there's a story that's been around for a while that there's efficient coding in the visual system and in the brain in general. So maybe what this means is you should represent things in a way where as much information is represented as possible with the resources you've got, and that would suggest, after some calculation, it turns out that suggests you should whiten the inputs. What does that mean? It means you want a flat power spectrum from all the spatial frequencies that are present. And if you look at natural scenes, they don't have equal power at all frequencies. Long wavelengths tend to have more power. So that would suggest a very specific procedure the retina ought to perform early in the visual system. And this was all driven by these principled ideas of efficient coding. So this is very appealing to me and many other people thinking about how the nervous system might work, and the idea that you could take a physicist's perspective and have principles to guide the way you think things might work, and then drive experiments. Markus found, though, that to the surprise of many people, including me, that story is actually accurate, but it turns out that it's not, the mechanism that the retina actually goes about doing this is not what you'd expect. It isn't just linear process. Filtering, there's actually a lot of nonlinear processes as well that I don't think anybody anticipated. So that was a really exciting surprise that came about from doing careful studies in the retina with this sort of physicist approach. I would characterize it that way as a physics chauvinist. But the bottom line is that he's come up with many surprising results that have given us, I think, a principled view of what's going on, not just a collection of facts. So it's been anyway. So that's one of many results that I was excited about from the retina from Markus's group. He then went on to work in other stages of the visual system, looked at Superiorcolliculus a little bit. He also had looked at olfaction, which is a very interesting complement because the olfactory space is very different than the sensory, the vision as a sense, the dimensionality of it, and other things. Coding schemes are different. He's also, in recent times, looked at behavior. I think he might talk about that today. I'm not sure. About mouse behavior and learning. He moved on from Harvard and has gone back to his in I guess 2012. Went back to Caltech, his original stomping grounds, and he's got a string of awards. I'm not going to list here. You can look them up. I'll give him a chance to give us his talk. It's going to be about dimensionless numbers in brain science. So, Markus Meister.
Thank you, Mike. Can you hear me? Is the microphone working? Hello? Okay. And thanks so much for inviting me. It's been a great day. So many exciting stories I heard. I think they're getting me all mixed up, but I'll try to remember what I wanted to tell you. So, not very good. Say again? The sound isn't very good. Yeah. Is this better? Any better? Maybe I need to button up. Try that. Yeah. So I've had a few questions throughout the day about the cryptic talk title, and the main purpose of the title is to allow me to choose what I want to talk about at the last moment. And in fact, you might have seen me do that a second ago. But I do want to spend the beginning of the talk on kind of more general comments about the discipline, I think particularly because this is a general audience. That will help, and somehow this theme you'll see is a good way to guide me into that. And then I'll give you short 10-minute vignettes about a few topics that I find particularly exciting right now. So dimensionless numbers, most everybody in the room knows what we're talking about. These are almost an addiction in the field of engineering. This is just the beginning of the first page of Wikipedia's list of dimensionless numbers. And dimensionless number comes about typically by taking one quantity and dividing it by another quantity that has the same units. So you end up with something that doesn't have units. And it's fundamentally a way of comparing the magnitude of two effects that are of the same kind, of the same type. Let's just be concrete. This is an example that's very commonly known. The Reynolds number is a way to characterize the flow of fluids around an object. It's essentially the ratio of the inertial forces, the forces you need to accelerate the fluid out of the way, versus the viscous forces, the frictional drag you experience. And in order to estimate that ratio, you don't have to be particularly precise about it. You just enter the basic parameters of the problem, the density of the fluid, the length of the object, the speed of the object, and the viscosity. And that gives you a rough ballpark measure of what's the relative importance of acceleration to drag. And when you do that, you find that a submarine has a Reynolds number of 100,000, and E. coli has a Reynolds number of 1 divided by 100,000. So these two organisms are 10 log units apart with respect to Reynolds number, and that tells you right away that propulsion of the submarine and propulsion of a bacterium have nothing to do with each other. They both use propellers, but even the propellers work differently. Yeah? Obviously, in the case of the submarine, frictional drag plays no role. In the case of the E. coli, that's the only thing that matters, and that simplifies the equations of fluid mechanics, as you know. And a great paper to illustrate the difference in lifestyles of bacteria and most everything else we know is this article by Ed Purcell from some time ago. So here's a way to quickly characterize a system and ask, is it in one regime or another regime? Oftentimes, these dimensionless numbers are named after famous personalities. One of my favorites is the Jesus number. This determines whether an organism can walk on water or not. So, for example, this Walter Strider has a Jesus number of 10, whereas ordinary humans have a Jesus number of about 10 to the minus 4. Okay, so in this way, you can get a quick characterization of a system and ask, is this possible or not? Can this thing walk on water or not? Yeah? But as you may know, dimensionless numbers have also been touted in a more fundamental way. Namely, it's been claimed that all the fundamental truths of a discipline are ultimately expressed in its dimensionless numbers. And so one way I like to illustrate that to my biology colleagues, and this is actually a good test whether I'm on target here, I imagine a situation where a physicist meets an alien and wants to impress the alien with everything we've understood about the universe. And you don't know what language to speak, so numbers obviously are the common language. I imagine the physicist would rattle off 1 over 137, 1836, and 1.75 times 10 to the minus 45, and the alien would immediately get a sense of how much we understand. Because, of course, one is the fine structure constant, the other one is a mass ratio of elementary particles, and still another one is the gravitational constant in suitable units. Now, I'm not making this up. I think we actually do talk to aliens that way. You might remember that the Pioneer 10 and 11 probes are flying around with a golden plaque affixed to them. And the instructions on the plaque say, remember what you know about the hydrogen atom. And then based on that, you can interpret these binary string numbers that we give you that tell you what the address is of the Earth in the universe. So there is an attempt to speak to aliens using these fundamental ratios. So in brain science, there hasn't been much of a tradition, I would say, of focusing on dimensionless numbers or using them in any way. But I think for us as brain scientists, it's useful to imagine meeting an alien and what would we tell that person about the object that we're interested in, and is there some way of thinking about it in dimensionless numbers? Or maybe in a more modest way, can we use dimensionless numbers for a quick characterization of processes in brain science that matter? And so over the last year or two, I've started to pull together some arguments for dimensionless numbers that I think each point to an interesting part of brain science that's either poorly understood or misunderstood in some cases. And you find that often when these numbers are either very small or very big, like the Reynolds number, that the system is in one extreme or another, and those are interesting extremes to look at. So let me start with a dimensionless number that is commonly called the dimensionality. And the question is, how many variables do you need to specify the state of the brain? And it turns out this question is actually getting a lot of attention these days, and has been, in fact, throughout history, I would say. And I want to just review some celebrated results from the 1950s and 1960s that speak to this number and give very different answers. So on the one hand, we had the experiments of Hubel and Wiesel, who recorded from single neurons in the cortex of a cat and stimulated the cat with many different visual stimuli. And they reported that each neuron has its own special receptive field. Each neuron is a special encoder of certain features of the visual image. And you move on to the next neuron, you find a different receptive field, and so on. And of course, the visual cortex contains many millions of neurons. And so one would say, based on this, that the dimensionality of the visual cortex is on the order of 1 million or many millions. At the same time, in another part of the world, we had Erich von Holst doing experiments on chickens. And he developed a method to very quickly insert a pair of wires into the brainstem of a chicken. I'll show you a little movie in a moment. And then he would just run an electric current through that pair of wires and watch what the chicken did. And in the movie, you're going to see the chicken standing on a platform, and down below the platform are these adorable old-fashioned dial indicators, and they're going to indicate the voltage applied to the two electrodes, and then you can watch and see what the chicken does. So let's see this movie. The movie is sped up just to save time. So here's a device that gets screwed into the ear of a chicken. Here's the assistant, the chicken on a platform, the dial indicators. And now watch what happens when the dial goes up. The chicken starts to peck the food. Every time the needle goes up, the chicken pecks the food. This chicken does something different. It pecks when the dial is low. When the dial goes high, it looks around for an imagined threat. This chicken again, when the dial goes high, it starts to look around, gets freaked out and runs away. This is a different chicken, does the same thing. Freaks out and runs away. So von Holst and his colleagues found that there were about 10 or 20 different regions in the brainstem of a chicken where they could stick their electrodes, and simply running current through those electrodes made the chicken engage in very characteristic behaviors. You saw it, right? Pecking, not pecking, looking left, looking right, running away. And so they argued that, at that scale of the brainstem, the dimensionality is about 10. There are 10 variables that I can control by running current through wires, and in that way pretty much cover the repertoire of chicken behaviors. All right, so there are two very different answers to the question then, right? So 1 million or 10 millions on one side, 10 or so on the other side. And I would say these discrepant views of dimensionality of the brain continue through the whole history of brain science to today. And I think which side of the divide you find yourself on really changes how you think about the brain and what experiments to do. Now, people don't usually carry their opinions around on a sign around their neck, so you have to figure out another way to spot them. And one way I tell my students is look at the cartoons that people draw at the end of their articles. So when they think they've understood something, what kind of drawing do they put down to summarize that? Yeah? And here's an example on the left. is a cartoon that appeared in one of my papers in 2007, and on the right, my close colleague Catherine Dulac, around the same time, published that cartoon. And so on the left here, every neuron is written a little bit like an electronic symbol, and every synapse connection between neurons is important, and the signals that are carried by individual neurons are important, and collectively they perform some function that doesn't matter here for this purpose. But you can see that the analogy to thinking here is electronics. You know, very high dimensional, there's many components, and they interact with each other in very specific ways. And of course, that analogy was not invented by us. There's a long line of thinking that argues that the brain can be understood in the same way as electronic or logical Boolean circuits can be understood. If you look at Catherine's drawing, it's very different, right? There looks to be some flow of information from the input to the brain to some nuclei, and in the left-hand drawing, the flow goes to the red nucleus, but somehow the flow to the blue nucleus is blocked, and in the other drawing, it's the other way around. And so here the analogy is, I would say, to a system of water tanks and pipes between tanks and valves that control the fluid flowing through the tanks, right? And again, this draws on a long history of this analogy. Konrad Lorenz described much of animal behavior using water tanks and valves and pressure as a metaphor. And so when I was young and naive, I thought, well, these are just two very different cultures, and only one or the other is correct. But increasingly, I think that both are probably correct at the same time, and it may well be that entire parts of the brain operate like a hydraulic system, and other parts of the brain operate more like an electronic system. And my colleagues and I at Caltech are trying to figure out how one can think about these sort of both the range of phenomena and how these phenomena interact. So the picture is that there's probably an outer brain, the part that actually has to interface with receptors and motor neurons in the outer world, that has these very refined receptive fields, and tens of millions of dimensions are needed in order to specify it, and operates at very large data rates. At the same time, there's also an inner brain that is probably where decisions are made, and elementary drives like hunger and thirst are expressed and are combined with memories in order to form decisions. And that part of the brain operates perhaps more like a system of tanks and pipes, and operates at much lower data rates. And so this is a kind of framework that I think can explain some phenomenology, and to me it kind of acts as a working picture for how one might choose some problems in brain science. So one problem that comes up here in particular is how do these, if this is true, if there are these different modes of communication between, or modes of operation, how do these different parts of the brain communicate? How do you get from the place where information is represented across hundreds of millions of neurons and at very high data rates to a place where only a few variables matter? And so we've been trying to turn this into an experimental program in a particular way, and this gets me to one of the dimensionless numbers I find interesting. I call it the sifting number. And it's related to this compression of data from the outer brain to the inner brain. The question is, of all the information that is available from the sensory organs, what fraction does a human being actually use for perception and behavior? And I would argue that that fraction is incredibly small. It's something like 10 to the minus 7. And here's an argument for that specific number. I'm looking at a particular task here. The typist is converting a handwritten manuscript into a typed manuscript. And you can ask how much information is available at the input here. So information available to the eye is on the order of a gigabit per second. There's a little back of the envelope calculation for how to get that. How much information does she produce? Well, we can measure that, how many keystrokes per second and what's the entropy of English. And it comes out to be about 20 bits per second. Yeah. So of all the information that the eye gathers and sends to the brain, she is able to select very precisely the 20 bits per second that matter, which is, I argue, is like finding a needle in a giant haystack. And in a way, this is sort of the power of the brain to be able to filter massive information flow by that huge ratio. And of course, it's an incredibly flexible filter because as soon as she gets up and walks across the room, she's again using only 20 bits per second, but it's a completely different 20 bits. It has nothing to do with letters on paper anymore. Yeah? And when she rides a bicycle, it's yet another 20 bits per second. Yeah? So in a way, this I find is an interesting phenomenon because that number is so small. We should really find out how the brain accomplishes this very tight filtering. An interesting consequence of this is, so I've argued that human behavior has an entropy of about 20 bits per second. It turns out that, whenever people have tried to measure that, they come up with the same number, somewhere between 10 and 40 bits per second. And so one consequence of this is that everything you perceive, every decision you make in a lifetime fits on a small keychain drive. Yeah? So if you take 20 bits per second as the rate of perception, human function in any domain, times lifetime of 100 years, and then it comes out to about 8 gigabytes. So we often talk about the brain as being this fantastically complex machine with enormous capacity and whatnot. I think when it gets right down to it, the capacity doesn't have to be enormous. I would argue that the things we have to remember from our experience can't exceed 8 gigabytes. And so, anyway. All right. This is just to set things up for discussion at the bar later on. This is, argument that, of course, leads to vigorous contention. Yeah. All right. How would you study something like that in the lab? So let me show you kind of an animal version of the sifting, of this very sharp filtering of information from the periphery. So this is our experimental animal. I'm referring, of course, to the small rodent down here that is escaping masterfully from the barn owl that is looking to catch it. Small animals like rats and mice are incredibly good at that, at evading predators from above. In fact, they're so good that they've driven all these ridiculous adaptations of the barn owl, the enormous eyes and big ears and the super silent wings and so on, simply by being so good at escaping. Now you can measure that in the lab by putting a mouse in a cage in an arena here. There is a shelter in the corner here. And we're going to project the image of a predator on the ceiling. It turns out you don't have to be particularly accurate. We're just going to put up a dark expanding circle. And I'll show you in a moment what happens when you do that. Okay, so the mouse immediately runs to shelter. We've measured the delays, about 200 milliseconds, so there's very short amount of time for this computation. It's very robust. Whenever you take a mouse out of the box, it'll do that. It doesn't require any training or accommodation or anything like that. So I think of it as almost like a visual reflex. It's also very specific. So if you try other kinds of stimuli, like an expanding white disk or a contracting white disk or just a dimming dark disk, you don't get any of these reactions. And so the system it's an alarm system. It notifies the animal of the likely predator, but it's quite selective to the features of the predator. It's, of course, what you'd want from an alarm system. You don't want it to go off willy-nilly so that you get interrupted all the time while you're foraging or mating or doing something else. So I would argue this is a circuit that allows the mouse to extract one bit of information from the visual scene, namely, should I stay or should I go? And if the circuit goes off, the animal runs to shelter. Okay, so we've been working a bit on the neural processing that might underlie this kind of filter to try and see how does the brain actually do it. Well, there are different pathways that are possible in the brain. The retina, Michael mentioned, the retina in the eye sends its output to two different visual pathways. One goes through what's called the thalamus to the visual cortex. In us, that covers a good fraction of the back of the brain. The other visual pathway goes to what's called the Superiorcolliculus. This is a parallel pathway. It's evolutionarily older. Fish have it, reptiles have it, and so on, whereas the cortex is innovation of mammals. And so the question comes up immediately, which of these two pathways is responsible for this kind of filtering, for the predator response? And there was lots of prior indication that probably this evolutionarily older pathway was responsible for these visual reflexes. I just want to give you some additional evidence. We've been working with mice that don't have a cortex. It turns out you can make a mutant mouse in which the neurons that would normally create the cortex die, or they never divide any further. And so this is an animal that has this is the normal mouse looking down on the brain. This is the mutant mouse. You can see that, there's nothing here. There's a little bit of tissue attached to the olfactory bulbs for smelling, presumably. But other than that, you're looking straight down on the midbrain and the brainstem. There's no hippocampus in these animals either. It turns out they're perfectly good mice. So the technicians in the animal facility have trouble telling them apart from the animals that have a cortex. And so in this case, we can ask, what do these animals do when the aerial predator arrives? Okay, so you can see the visual reflex seems to be working just fine, but there's something wrong with the endpoint of the behavior. They don't actually make it home. Yeah? And I have some ideas why they don't get to shelter, but all the visual processing is okay. And in fact, we've shown that they're also equally selective. They don't respond to these other stimuli. Yeah? And so here's an animal that doesn't have one of the major visual pathways, so presumably the information is processed by the other pathway, the Superiorcolliculus. So let me tell you in a few minutes some results from sticking electrodes into the Superiorcolliculus to try and understand how this processing develops. So Superiorcolliculus is a part of the midbrain. It is a layered neural structure. The whole top to bottom here in the mouse is about 1 millimeter, just to orient you. The top of the structure gets input from the retina. These green things here are axonal fibers that come from the retina and make terminal arbors, connect to neurons up here. The bottom parts of the structure are actually described as being more motor-related, movement-related. So if you do a von Holst type experiment and inject current into that location, you can get the animal to do entire orienting behaviors away or towards an object in the environment. So we wanted to know what is the transformation like going from the top of the colliculus to the bottom of the colliculus? What happens in the neural signals in this structure going from an area that's close to the senses to an area that's close to the motor output? And so experimentally, we use these multi-electrode arrays. They're basically silicon prongs with many recording sites on them. They are about 1 millimeter long, which is nicely matched to the height of the structure. And so it allows us to simultaneously observe neurons in the different layers of the colliculus. And I'm just going to give you a quick sense of how different the responses at the top and the responses at the bottom of the structure can be. This is sort of a quick summary. I'm going to compare one neuron in the superficial Superiorcolliculus, the upper layer, to a neuron in the lower layers. And what I've plotted here on the left are the different stimuli that we use to present to the mouse in order to see how these neurons respond. And each of these traces is a report of the firing rates plotted vertically against time plotted horizontally. And time, this whole axis is 20 seconds or so. So what happens is that every time there's a pink bar here, we present one of these expanding dark disks in the superior visual field. And this neuron fires a burst of spikes of 50 or so hertz, and then goes silent again. And then it fires again and goes silent again. If we present an expanding white disk, the neuron does the same thing and again responds with bursts of spikes. Contracting dark disk, the neuron does the same thing. Contracting white disk, again, the neuron responds strongly. So you can see that this neuron responds to many different kinds of stimuli, unlike the whole animal. The whole animal performs this escape only for the expanding dark disk. Now, in the deeper colliculus, you find neurons that look like, whose responses look like this. You can see that this neuron fires a big burst of spikes in response to the expanding dark disk, but not so much in response to all these other stimuli. Another weird thing about this neuron is that it fires only the first time. So when the stimulus repeats again, you get almost no firing from this cell. It's as though there's some kind of habituation, and I'll have more to say about that in a moment. And then a third difference between the top and the bottom is the neurons at the top, they are sensitive to these expanding disks in only a small region of the world, basically a few tens of degrees around their receptive field, whereas the neurons in the deep superiorcolliculus are responsive over the entire visual field, 100 degrees of vision apart. And so there is a clear transformation then from the top to the bottom to a representation that is much more selective for this predator stimulus, at the same time is invariant relative to where the predator arrives from, and then has this interesting habituation property as well, this interesting form of memory. Let me say a little bit more about this memory. You might think the simplest hypothesis is, oh, the neuron is just getting tired of firing. So if it sees a stimulus that drives it strongly, then something happens, something gets exhausted, and it can't respond anymore. So that's not the case. So if you do an experiment like this one, where the stimuli appear randomly at different locations, we can now ask, how strong does a neuron fire, depending on whether that location has already happened once or not. And that's what I'm plotting here. Vertically is the response of the neuron. Horizontally is the sequence of trials in this random location experiment. And you can see the dots that are connected with each other are the identical stimulus, meaning the same location being stimulated. All these lines point downward, so that tells you that the second time the stimulus produces a much smaller response than the first time. But interestingly, look at this example for it here. In the time that the neuron's response to the blue stimulus obviously declined by a factor of 3, it still produces a very strong response to other stimuli developed at other locations. So the habituation is not a form of fatigue. It's not that the neuron somehow is exhausting. Instead, it has a selective memory for which stimuli have occurred and which others have not occurred yet. So there's some kind of stimulus selective memory in the system, and the neuron is responsive to novel stimuli and not to familiar stimuli. Well, what does novel and familiar mean? How long are we talking about? Eventually, of course, the memory must disappear. It turns out the memory is quite long. It lasts on the order of 2 minutes or so. So if we present the same stimulus at different time intervals, we find that one has to wait about 2 minutes to recover the sensitivity. Okay, well, this starts to sound like memory formation, perhaps. The textbooks say that the hippocampus is important for memory formation, but not for this memory. This persists in absence of hippocampus or cortex. Again, using these cortex-less mice, we can find that the lower superiorcolliculus has the same level of habituation to stimuli, or if you can think about it differently, a preference for novel stimuli as in the normal animal. We've started to think about neural networks, circuits that could produce this kind of sensitivity, and I would say this is at a very early stage where we're kind of trying to create some models that can be used to plan new experiments. In this model, is that some neurons are selective for the local looming stimulus, but only in one location, and they become selective through a particular input circuitry. And then there's a second stage where neurons pool inputs over large distances, and that would lead to this sensitivity over a great part of the visual field. And if, in addition, one adopts some kind of plasticity at these synapses here, one can imagine modeling the appearance of this novelty detection. Again, I don't want to spend too much time on this. This is quite speculative, but it helps us in phrasing experiments that will distinguish between one possibility or another. But let me just summarize this part as I think this is an occasion where we might be seeing the outer brain and the inner brain communicating. We might be seeing this transition from a representation that's more at the raw data level of the retina, meaning very high dimensional, lots of things are being represented, to a central representation where the filter has been applied and only a few things make it through the filter. Okay, let me switch to a different topic. This is another dimensionless number. The topic is very different here. We're talking about biophysics now. It's about a technique that is being developed over the last few years in response to the exhortation by President Obama's Brain Initiative to develop new tools that could be used to stimulate and modulate the nervous system, ideally using methods that we haven't even thought about before, like physical principles that aren't part of the repertoire yet. Now, many of you know about this fantastic success of optogenetics, the ability to modify neurons genetically so they can be stimulated with light. It's led to a minor revolution in the field. And it still has a drawback that you have to somehow get the light into the brain next to the neurons that you're trying to stimulate, and most animals are not transparent. And so the idea was, what if we could use magnetic fields? They pass through the body almost undisturbed. If one can couple a magnetic field to the activity of a neuron, that would be a great way to develop a new tool. So in 2015, 2016, several papers appeared that claimed to have done this by constructing a new type of protein molecules. And in particular, they took a membrane channel. This is a protein that allows for ion conductance through the membrane and can excite neurons that way. Link that membrane channel to a protein that's called ferritin. Ferritin is like a spherical shell into which the cell stuffs a lot of iron atoms. It fundamentally wants to get rid of the iron and puts it into this shell, encapsulates it, and then gets rid of it. So the idea was, if we can link this protein that has a lot of iron in it to a protein that controls the conductivity of the membrane, then we can use a magnetic field to pull on the iron, and in that way maybe yank this channel open so it passes ions. That was one approach. The other approach claimed that they were using alternating magnetic fields to heat up the iron in this ferritin by basically flipping it back and forth very frequently and creating heat that way, and then the heat would spread to the ion channel, and because it's a heat-sensitive channel, it would open and excite the neuron that way. So this was all very exciting. It got a lot of hype in the press, and you can see why, because if it were possible, you'd suddenly be able to control even the human brain by simple magnetic fields. It'd be fantastic. So what's the problem? So the problem is dimensionless numbers. So some time ago, I calculated what would the force actually be that you can exert on this ferritin with a magnetic field, and how does that compare to the force you need to open an ion channel, and the ratio is about 10 to the minus 9. And I assure you, I didn't need any higher physics here. Ed Purcell's Berkeley physics course is sufficient to do these calculations of interaction between a paramagnetic particle and a magnetic field. And similarly interesting, if you ask how much heat can you develop by magnetic heating of ferritin, and how does that compare to the heat you need to open an ion channel, the ratio is about 10 to the minus 10. And so I wrote this paper 3 years ago saying it's very unlikely that things happen the way they were described here, and my prediction is that this isn't going to replicate. And it led to a lot of discussion and dispute and whatnot, but so I'm pleased to say that you can use dimensionless numbers to predict the future, because we're now in 2019, and you can find on the bioRxiv 2 of these articles. The third one will come out soon in Nature Neuroscience, that report that you can't replicate these effects. Fundamentally, these articles, the 3 articles in the series, each one of them goes through much greater lengths than the original articles reporting the effect, and reports that they can't make this work. You might say, why did it take 3 years? It took 1 year to do the work, and it took 2 years to convince Nature Neuroscience to publish it. And so with some luck, we'll be able to see it in the next few weeks. Anyway, I've been told by Julius Zhu that he also has colleagues in China, that there are on the order of 100 research groups there that have attempted this because of the fantastic promise that it held, and none of them is making any progress. So there's been this sort of deafening silence on the on magnetogenetic front. So it's an argument that, it would have been useful to do this calculation ahead of time. Probably could have saved a lot of postdoc years, a lot of graduate student years, chasing this effect down that probably is going to end up being a result of human error. Mike, you told me to finish up, right? Okay, all right. So I'll skip the details because we're among physicists, so you don't need to know this. Oh, you already know this. Okay, all right. So I'd rather move on then to another little vignette about a dimensionless number that I enjoy. It's the answer to the question, how many times am I going to have to tell you this? And if there are any teachers in the room, you're probably well familiar with this. So I'm going to define the teacher number as the number of trials an animal needs to learn a new task. So let's see, we've already seen some one example of this. So I'm going to focus now on mice or rodents in general. And let's look at a few things that rodents actually do in nature. So this is one, avoid aerial predators. And we've already seen that the teacher number for this task is 0. They do it the first time, and you can understand why. There is not a lot of opportunity for practice. Here, a mouse finds out that an insect is delicious food, and the teacher number for that is 1. I can tell you about that a little in detail later, but we basically take laboratory mice that have never had to work for their food. They get cookies their entire life, and we force them to chase live insects. And it takes a long time. It can take 10 hours for the animal to finally consume the insect, but the next day it takes 2 minutes. So something happens in the brain of the mouse that changes its attitude about live insects from one experience. Here, mice live in burrows, and so they need to find their way, of course, around sometimes complicated burrows. And I'm going to argue that the mouse can learn a complex maze on the order of a few experiences. So you might wonder, why do mice have such a lousy reputation? Because at least all of my colleagues have heard many complaints about how stupid mice are, and the rats are so much better, and monkeys are much better, of course. So mice have a terrible reputation when it comes to learning. And I think the reason is that many times when we try to teach a mouse something, it's an abstract task like this one here. So this is these are experiments from my own lab, but you'll find much more of this in the literature. Here, a mouse is in a box, and it has to poke its nose into some port in the center, and then a light comes on the left, and the mouse has to go to where the light is and poke its nose in here. And if the light came on the right, it would have to go to the right and poke its nose in there. So it really has to only distinguish two things. It's a one-bit decision task, right? So you can't make decisions simpler than that. There isn't anything less than 1 bit in a decision. So, and you might think it's so trivial that the animal should just pick it up right away. But instead, here are some curves of performance, meaning fraction of correct responses versus trials. This is, again, from my own group. And you can see that it takes the animals thousands of trials to gradually improve their performance, and they never get particularly good at it. Here's a much larger data set from the Carandini and Harris groups at UCL. And 100 mice were trained here in the acute task where the animal has to turn a steering wheel, depending on whether the stimulus is on the right or on the left. And again, they learned this rather slowly. The solid curve here is the median mouse, and it saturates after about 10,000 trials, and it saturates at about 80-some% correct. Yeah? Now, 80% correct is not good, right? You get 50% for not doing anything. That's, yeah? And so 80% is like not even two-thirds of the way. Okay, so the two important questions here. Why do they take so long to learn this, and why do they never get particularly good? Well, it turns out that this same mouse, one of these mice here, if you test it in a different task that seems much more complex, it can actually learn very quickly. So here's a different task. We built a maze, a literal maze, a warren of binary decisions. The animal can enter the maze from here. Its home cage is attached here. I haven't shown it, but the animal can enter from its home cage, go into the maze, and find water. The animal is thirsty, and there's a water port up here, this location. Now, to find water, the animal has to make 6 correct decisions in a row. Any other sequence of choices will lead it astray. So I just want to show you the very first time this animal enters the maze. This is what it looks like. By the way, we're taking a video from below, so you can see the little foot pads. It's kind of cute. Also, the maze is, the corridors are tight, so the mouse looks more like a snake. But, so you can see what it's doing. It's really exploring this environment, grooming itself a little bit, and looking left and right, and so on. And this will go on for a long time. I'll tell you how long. Yeah. All right, so this was its very first entry. After about an hour in the maze, the animal for the first time discovered that there's a water port. Now, when it collects a few drops of water, it then has to return along this green route. It can't go back the way it came. There's a valve there. The animal can't return. So it has to go back through a different route and start over to get more water. Yeah. All right, after 2.5 hours, the animal for the first time executes all 6 decisions correctly, so it walks straight to the port. And then a little while later, the animal does this. Like I said, it gets only a few drops of water, then the port shuts off again, has to come back, but it's still thirsty, so let's go again. And so on.
Why doesn't it go the civil way?
Say again?
Why doesn't it go the civil way?
There are one-way valves in the way. It can only pass.
What do you mean by one-way valve?
Check valves for mice. So it's a little trap door that only opens in one direction, not the other direction. Yeah. Anyway, so you get the sense by this time, by about 10 experiences, the animal has learned this task perfectly and will do it as often as you want, or as often as it wants. All the time. There's a lot of interesting phenomenology. It turns out that oftentimes, even after this level of perfection, the animals will just go in the maze seemingly to explore. I mean, by this time the animal knows where the water is, and yet it will engage at times in a systematic search of the entire maze to see if something new happened. I mean, I'm just hand-waving, of course, but there's a lot of interesting phenomenology in these movies. So anyway, the question, I forgot to mention, this is also one of these animals that doesn't have a cortex or a hippocampus. So you don't need a hippocampus to find your way through a maze. Anyway, the question is, what is this contrast? Yeah, we're now faced with a situation where mice seem to learn incredibly slowly and be very poor, to very poor performance. In other cases, they learn fast and to high performance. And so here's sort of my view of the history of this particular field, is that in the old days, animal psychologists used real mazes. In fact, this first one was constructed following the layout of the gardens at Hampton Court, of course in a smaller scale. Then Professor Small watched rats make their way through this maze. Later on, a lot of interesting work by Pullman, who kind of simplified things and ultimately decided, look, the elementary decision act in a maze is whether to turn left or right, so why don't we just focus on that T-maze and try to study the elementary decision act. And then it was decided that this is still tedious because the animals walk out on this side and we have to bring him back to this side, so let's keep the animal in a box so it can't walk anywhere, and instead we just make it turn left and right with this, with these response ports. Yeah. Okay, so a classic case of scientific reductionism: you take a phenomenon from the real world that is exciting and startling, and you bring it to the laboratory and trim it down and try to control all the conditions and remove complexity so you can experiment with it. But then, something obviously has been lost in the process of this reductionism. Because we're now faced with animals performing trivial tasks for 10,000 trials and getting lousy performance for it, whereas we know that they can perform on complex tasks with high performance after just a couple of hours, or as we now know for animals with a cortex, 10 minutes. So what is the resolution? To me, it sort of harkens back to this idea of a filter between the outer brain and the inner brain. I mean, I'm sure we have one, but the mouse has one too. And this filter by which the animal extracts relevant information from the surroundings, I'm sure it's very adaptive and can be modulated by learning and whatnot, but it's not infinitely adaptive. And my guess is that what we're asking this animal to do just isn't part of what it means to be a mouse. It just doesn't fit into its perceptual filter, whereas this sort of thing is obviously part of its natural activities, and so if you challenge it on its own ground, so to speak, it can perform for you very well. Yeah? This, I tell my students, is a little bit like trying to teach students echolocation. And, we have an echolocation expert here. So humans, in principle, have access to the information you need to do it, but, there's one person who's learned to do it. So I feel like we have to imagine what it's like to be a mouse, and my feeling is that these tasks are a bit like asking us to do echolocation. So more importantly, I feel like the fact that this teacher number is so much bigger in this case than in that case, it's a bit reminiscent of the bacterium and the submarine. It seems to me like these two tasks are really asking the animal to do completely different things. And seeing this big difference in teacher numbers, I would argue that this form of learning may well have nothing to do with this form of learning. It's quite possible to me that just like the bacterium is propelled differently from the submarine, we're really studying two very different processes by these types of experiments. So to summarize then, I've given you a small selection of dimensionless numbers. I find this an interesting way to get into an area where there seems to be a big unexplained effect, or at least a large effect that might help you gain some insight into the topic, in this case, the topic of the brain. And finally, let me just thank my students and postdocs who've obviously done all this work, and also collaborators at Caltech with whom I talk a lot, and also donors to the institution that help me or allow me to think about things that wouldn't be fundable in any other way. Thank you.
We've got time for some questions.
That's one of the vignettes I was telling about. Well, so, very briefly, I told you about the sifting number that's very small, 20 bits per second total bandwidth for perception. Our argument is that if someone is blind because the light stops working, you could try to bring the information into the brain through another route at the 1 gigabit per second level, and a couple of companies have gone bankrupt doing that. Or you could say, ultimately, this person only needs 20 bits per second. Let's give them the 20 bits per second that really matter. And so we're developing some visual prostheses that use natural language, basically, to convey the visual scene at 20 bits per second, and try to form some internal mental representation of that. The inspiration was actually speaking to a blind person about what they like to do, and he said he really enjoys watching movies. And of course, we asked, well, what movies? He said, more and more movie theaters have audio narration of what's going on the screen. And you get it beamed into infrared receivers and headphones. And he said, you can try this out yourself, actually, if you listen to Netflix movies, they have some audio narration. And he said, for a while, it's confusing, and I have to adjust to it, but after 10 minutes or so, I find that I don't hear the voice anymore, but I see the characteristics going on on the screen. And so we're trying to tap into that idea. Yeah.
So are you saying that we should be going with teammates with them? Because teammates is not like a make a left and a right hit, spatial paths, and it can be very easy to solve, right? So it's not, you can't
guess is, and I think there's some evidence for it, that if you make the animals walk and get them to communicate their decision, even give them a virtual reality illusion of being in a corridor and taking turns and so on, you already get them more engaged and you get higher performance. So I think the real maze is just kind of one extreme end of that. Animals love it. I mean, so we give them free access to the maze. They can go in and out as they wish. They spend about half the time walking around in the maze. So the home page must just be infinitely boring. So nothing good ever happens. They still seem to get something out of the novelty.
You're basically saying that study what the animal will do that, right? Because one thing I'm wondering about your mice is that, so if you have a bit more of a complex thing, where they can get from the beginning to the end, where they can just move around, really actually you mentioned a lot of variation there. You try to optimize around it, to focus around, or they just want to come around and the new point and
Yeah, we haven't quite engaged. We've started, but we haven't engaged that topic yet. This was actually something that homeowners were interested in. With the animals develop an internal representation of the space that allows them to do computations like what if this path is blocked, what's the shortest path? Another way. And so we could follow up on that as well. I should say that those psychologists were all very clever. And so they asked a lot of questions already that they weren't able to answer at the time. Now with the tools we have we were talking about this, right? Video recording and animal tracking and documenting and reinforcement learning models of behavior and so on. I think one can bring a whole different approach to quantitative analysis of these behaviors.
There's a question back there. A while ago.
Yeah, it's interesting. So we've worked with those all time. So people were visually aligned and people were aligned on verbs. And the experience of the visually aligned, it really does matter. So they have a different intuition about the geometry of space than the visually aligned. The particular device or approach we're taking to this prosthesis actually doesn't distinguish between the two. I think it works equally well. Bring me here again, I'll give you the other top. So is there a principle one could draw on to predict which kinds of decisions perhaps are learned easily versus yeah. I think Michael said earlier, the ecology of the animal as a guide to what it needs to do and what the kinds of, as a result, what kinds of decisions perhaps are learned more easily than others. I think that's a principle and also has a long tradition. I think it's important to ask yourself, what will an animal actually have to do in nature? And the kinds of things that they take 10,000 trials to learn, I just fail to believe that they matter to an animal's reality. I mean, unless you're a chess player, Tiger Woods, Roger Federer, antenna serve is not active. But in a way, we admire these people because they're so far out of the spectrum, right? Because they're performing at a level that is unnatural. And so I think if the emphasis is on how to have a survive in the wild, my guess is that with a few exceptions we know about, there's 1,000 files really.
Marlon?
So I'm going to be a devil's advocate here, because I think you're mapping a sort of sensory representation done by neurobiologists, which use a high dimensional space and therefore have lots of bits of information. You're mapping it onto psychology. You know, all I have to do is decide if I'm moving left or right, which is a low dimensional description of what's going on. So in the example of the type tips that you have, there's of course a tremendous amount of information that's being generated in the brain that has lots and lots of bits of information, so that type is to be able to hit the keys correctly, right? So motor outputs are very high dimensional representations of what's going on, and behaviors have a lot of motor outputs associated with them. So I think that it's a you know, just by your describing it, you only have to explain language. It's a little unfair for saying what the brain has to do, what the nervous system has to do with that sensory information.
Well, I'm in awe of what the nervous system does, because at the end it's really just about 20 minutes. I know there are lots of nerve fibers and lots of exponential nerve fibers, but in the end, she just, you know, every time she hits the E key, she hits the E key.
And hitting that E key requires many, many synapses, lots of muscle control that's coordinated, right? That requires lots of information generated in motor cortex, so it's done.
There's no information generated.
That's
That's
right,
here I'm not saying it doesn't happen. So it really is a compression process. Right? You go from full information she has about the visual image to ultimately being able to get it to survive.
Aren't you talking about like the dimensionality of information? You're saying I am dimensionality of the brain and also motor aspects. So you can actually learn that language not how the brain
So the people that measure the human behavior in other ways as well. Maybe we have to be a sight reading. People are trying to make real thought. Like how many different thoughts can you actually imagine happening? But 20 bits per second sounds like a small number, but think about it. 20 bits means 1 thousand out of a million. So the claim is that even if you think about your internal, your inner life, your thought life, that contingent on everything you've thought about up to now, there are a million possible trajectories in the next second. So thinking about my own internal operations, I would say that's more that's probably much more predictable than so that's what we're talking about. What is the diversity of thoughts, or diversity of perceptions, or the diversity of movements that human beings can generate?
Maybe one more question.
So while we belabor the analogy of the hydraulic and electrical circuit, I'm wondering, is there any real life structural chemistry saving for a hydraulic and an electrical circuit integrated in such a way that for an interesting property actually emerges in that which is like that?
Process control, whether refinery.
So, one of our colleagues that we're working with is Pietro Perona. He thinks about machine vision and a lot about control of autonomous vehicles. He says there's a similar problem there in that when you have at the periphery of the vehicle, you have enormous numbers of pixels of visual data, of sensory data. You also have complicated effectors for the steering of the vehicle. But at the core of the algorithm running the autonomous vehicle is something rather simple, like an instruction
Do they have anything else?